I also appreciate the opportunity to talk on some work we did on thermal desorption tubes and exhaled breath data. I have no conflicts of interest, and all of these are my opinions. Okay, so a little bit of backstory. These data were acquired coming out of the pandemic. And so not knowing what our ability to sample humans was going to be, we were starting to brainstorm ideas of research we could do independent of the human. We discussed through teams, and we started to slowly trickle back into the lab about things that would be worth investigating. And so we talked a lot, and we landed on investigating the background from TD tubes. And then we have some anecdotal evidence when we do calibrations surrounding the variable flow rates among TD tube types. So in the background, traditionally we would run blank tubes to analyze within samples to ensure our system was clean. And then in the end, we would evaluate, room blanks and those kind of things to determine the features of interest. Were not found, but we never really truly integrated or had a really nice plan for what we were going to do with those blank data. And then, like I said, the flow rate portion comes from evidence that when we calibrated our pumps using flow rate monitors, we would notice it was variable depending on the TD tube. So we wanted to explore kind of these two scenarios. And so I'm going to talk about background first. Traditionally, for offline analysis, exhaled breath contains very low concentrations of the majority of the analytes. So pre-concentration is usually necessary. Usually this is done through thermal desorption tubes, but there are other ways to do so, such as FEMI and headspace, if you really want to get into it. The two most common that we have in our lab are generally based on 10x, so we use 10x TA. And then the Carbograph 10x dual bed, which is a biomonitoring tube, both of which are from Marks. Each tube has its own pros and cons, and again, these aren't limited. These are just what we primarily use in our lab, seemingly what a lot of researchers are using. But 10x really doesn't retain the low molecular weight compounds great, and the biomonitoring have humidity issues. So it's kind of tailored to what you want to use and how you're going to mitigate the issues. So in our lab, we feel that to properly evaluate exhaled breath data, we want to consider all the facets that impact the results. And so, like I said, historically, there's been a lot of work investigating storage on tubes and in bags, background handling of exhaled breath data. But little has been done to evaluate the actual tubes themselves. And again, the references that I have are not exhausting. There's other literature out there if you go looking. So what we did, so this is a little arrow guy down there. Exclamation point is a photo of my instrument, which didn't make it apparently. So what we did is we took over 200 of each 10x TA and biomonitoring tubes randomly selected from our stock of about 500 or more of each. And then we took 15 adsorbent free, so stainless steel and inert coated empty casings. And we ran them using our standard GCMS method without internal standard. And all of our materials were from Marks. And so this is kind of a summary of our method, and you can picture what a TD and a thermo GCMS single quad looks like. Yeah, sorry, I didn't make it. So if you look at the data, it looks something like this, where your lighter gray is your your biomonitoring tube, and then 10x, and then on forward in the darker is your empty casing. And so you do find some backgrounds related to that. And so just to confirm that it was coming from the tubes and not necessarily our system, we actually ran the controls. We ran controls for the GC trigger to ensure that our column wasn't contributing to these results. And we also ran a trap fire, which is the secondary desorption, and neither of which showed any background in those results. Okay, so to evaluate the background, we took 3 times the signal to noise, approximately around 10 minutes. All of the tubes roughly had a background level around there. And we set that as the minimum signal for feature extraction. Um, we extracted all of the features, retention time, aligned them, and then from those features, we went through and manually did spectral matching to the NIST library. There's the GC and trap only. So these are the controls to ensure that it's coming from not from our system, but actually from the TD tubes. And so what we found is the biomonitoring 5TD, the dual bed tubes, have the most number of features using the criteria that we used, but again, both adsorbents contained background features, whereas the empty features were much less. So if you look at the tentatively ID compounds, you also find that the biomonitoring tubes with the dual bed actually have the most abundance of those background features as well compared to 10x and their tubes. And this kind of makes sense. You're grabbing a wider range of volatiles. So it's not really that shocking that this is the case, but we wanted to go through and see what it was. And so to kind of put this into perspective within the exhaled breath scenario, we went through and used these two really pretty well-known review articles and looked for things that we found in our background that also overlapped with things that were found in breath in these review articles. And we found quite a number of background compounds were reported in the literature in these reviews. And so that made us think about, going forward, what would we how would we evaluate whether there's an actual impact of the background or whether these are just kind of artifacts? And so we wanted to evaluate it in exhaled breath. So what we did is we had 10 individuals provide a rested breath sample into an alt bag and using lower airway breath. And we transferred 550 mils on both 10x and the biomonitoring dual bed tubes and ran them our GCMS. And then the exhaled breath data was compared to the abundances from our background study for those previously reported in the literature. On the left, you see the results for 10x, and on the right, you would see the dual bed. And what you find is, depending on the compound, up to 70% of the signal could be contributed to the blank data or the background data. And then so additionally, there are in the biomonitoring tubes some features such as toluene and phenylhaldehyde that do not show any statistical difference from blanks to breath. So, there seems to be potential for impact of your background data or your background samples and the TD tubes to plausibly impact your exhaled breath data. And so we feel that these results suggest that true evaluation needs to be done. And so I'm part of the standardization community within the IABR. And so I would like to kind of highlight that these results, some sort of standardization about how to handle breath and background data needs to be addressed. So that was kind of all of the background results that I had. And so going forward, I'm going to talk about some of the TD2 flow rate data we acquired and some exhaled breath that's related to that. So again, anecdotally, just by calibrating, we used Defender, which is our flow rate monitor. We use a representative tube, and then our multi-rate pump is how we transfer volatiles from our bags. So we use a representative tube to calibrate for flow rate when we transfer from a bag. But since we want to know what the flow rate is, we tested over 200 tubes of each 10XTA and the dual beds, and we measured the flow rates of those tubes. And what we find is there's quite a range in flow rates if you measure them. The image is of the mean flow rate over 10 individual measurements. for each of the 200 tubes, oldest being oldest numbers tube on the left, newest tubes on the right. And there's quite a range there, and it can vary and quite a bit. And so we wanted to investigate more about what could be contributing to this flow rate. And so the real question is, why does this matter? And so when we use a representative tube to calibrate for flow rate, we assume all tubes are equal at that point. And if they're not equal, this could lead to unequal loading. And so if you, for example, if you use the lowest flow rate of 220 mils per minute for a representative tube, and then your actual tube that you're sampling is one of the higher flow rate measured tubes, you could oversample your bag by over 15%. So there is a potential that this could truly affect your loading, because all of your data is related to consistent loading. So all of the TD tube data is assuming that you loaded the same volume on every tube. And so there is a plausibility that this could affect your data. But I have some results where we're going to talk about it. So I have over 1,000 tubes, and so actually, determining the flow rate for each individual tube isn't really feasible, and at least in my lab. But it would be nice to know whether there is an impact. And so what we did is we wanted to know first, does the mass of the tube correlate to the flow rate, i.e. more packing, harder to get through, less packing, harder to get through. And so in the top left, you can see the mass measurement for each tube. And you do see some variability, but if you plot the flow rate by mass, you do not see a linear correlation, while it may or may not be a linear type of analysis. Based on our data here, it does not seem to be linear. And we also measured just empty tubes, and there doesn't seem to be a large amount of difference between the inert and the stainless steel, suggesting that we don't feel that it's necessarily related to the inert casing or the stainless steel casing, but probably related to the actual packing as the differences in the masses. But we do not see a correlation between TD tube mass and the measured flow rates for those tubes. So we have a really nice tracking system in our lab where we track the number of reconditioning cycles. And so we went back through our system, and we noted the number of reconditioning cycles. And what you can see is there's definitely a downward trend, which would be expected. When you go from oldest to newest, you should have more reconditioning cycles in your oldest tubes compared to your newest. But we do not see a linear relationship between the number of reconditioning cycles and the flow rate. So that doesn't seem to be what we're finding contributing to the differences in flow rates either. So again, the data indicate that flow rate isn't based on age or the mass or the number of reconditioning cycles. So we hypothesize that it is how it is actually packed within the tube, i.e. Is it kind of stuffed in there where the flow is harder to get through versus more loosely packed within the tube? Well, the question was, but does it really matter? Right? If you see this variability and you still get consistent results, or you get results all over the place, you really need to know. So that's what we set out to do next. All right, so to test the variability of the TD tube flow rate with exhaled breath, what we did is we picked 15 of each tube type that were high flow rate, medium flow rate, and low flow rate based on the results. So the extremes in the middle. And what we find is the attributes that we checked, so mass and reconditioning cycle, aren't significantly different, but the flow rates that we measured were for each condition. And so we use those to investigate the impact of flow rate with exhaled breath. So we had 10 participants, and they sat in a relaxed position. They provided a breath bag prior to ingestion, and then we had them have a wintergreen Altoid and let it dissolve on their tongue. And then after 5 minutes, we had the participants provide an additional lower airway breath bag. And then we took 550 mils on each low, medium, and high tubes for each 10X and the dual bed tubes, using our standard protocol where we would use pick a random tube and use that single flow rate measurement to transfer the volume and do the calculations. All the data were run on our TDGC-MES, as I suggested before, and we did a targeted analysis for the MINT compounds, which we ran MINT to get the volatile headspace, and we found 2 compounds were most prevalent in the Altoids. But then we also monitored acetone and isoprene, and then we did our kind of global feature extraction, alignment, and global analysis. The data show that there is variability in the abundances, but they're not statistically different. So these are the top right is the 2 MINT compounds, and you can see there's some variability, especially with methyl salicylate. So we believe, compared to the other peppermint studies that had been performed in the literature, we didn't have them ingest a capsule, so they had an oral breath mint, and so there was variable time in the amount that it took for that mint to dissolve, allowing to potential off-gassing, which is why we believe that there's some variability here, but we don't see anything statistically different in the mint-related compounds based on flow rate or TD tube. Similarly, we monitor some historically endogenous compounds, acetone and isoprene, and we see a very similar result where there is some variability here. We believe that's just endogenous variability. That's pretty well documented in the literature, but we don't see any statistical difference based on low, medium, and high flow rates for either compound or either TD tube type. And so we did a global analysis, and I think we monitored them from 46 features out of our data, where we excluded the MINT compound. So everything retention time earlier than the MINT compound, which I believe is 18 minutes. So that's the majority of the things we normally see. And what you can see is on the left would be your 10X, and on the right is your biomonitoring. And what you can see is a high amount of overlap, independent of pre-post mint and high, medium, or low flow rate. And additionally, what you in the 95% confidence interval, and additionally, what you see is there's a relatively low amount of variability accounted for by PC1 and 2. And so we believe that, or we hypothesize that a lot of there is no one distinct source of variability, which you would believe that if it was based on flow rate, you would see more of your variability accounted for in the principal component analysis, suggesting that your variability is probably dispersed among all your principal components, suggesting that these data are very similar to each other, both in a targeted and untargeted fashion. So interestingly, while we do see differences in flow rates, the evidence suggests that there is minimal impact to exhaled breath results. So with that, there's also some caveats. We tested a small group of people. We used a random tube, which is for our flow rate calibration. And so there is some plausibility that if your random tube was on the lower end, you might actually see some samples that the variability is large enough there in your flow rate based on your calculations for your calibration that it would allow you to plausibly more impact your results. But based on the results I have here, there does not seem to be a large effect. So one of the things that I wanted to go back to with the background data is We tested our systems. So I believe that there should be, for people to use, should evaluate their own systems and their own blanks and their own background to see how they can contribute to their exhale breath results. While the TD tubes show variable flow rates, exhale breath doesn't seem to be impacted. And we're not totally sure why. We also, within this analysis, used standard bags as well. So we have standard data as well, and those did not show any difference based on flow rate either. So we're not really sure what is going on there, but we have some ideas, and we're going to try to evaluate those going forward. So we are always looking for ways to improve our sampling, and Christina Davis's group came up with a sampling device that does real-time flow rate. And so that is a viable option, which we're investigating something similar. We want to continue working on standardization and evaluating things that impact exhale breath collection and analysis. So majority of the work I presented here was done by a researcher in my lab. Her name is Anne. She did 95% of the data collection here, and then Heidi was actually a United States Air Force Academy student I had in the summer, and she did quite a bit of collection as well. But they did a majority of the analysis and collection, and I just get to talk about it, which is really nice. But then the rest of my group is also included. They have all helped in one way or another. So this is a plug. I have a special issue on exhale breath standardization and contamination. For more information, please go to the JBR website. And if you have some data that might fit within the scope, please submit it. It is open until the end of the calendar year. And we would love to have anything related to standardization be submitted and considered for publication.
Excellent, wonderful. Thank you so much for that, Sean. Great presentation. Great to see the data there. I was going to kick us off. I thought the data showing the levels of VOCs in background relative to VOCs that are on breath, and then showing often those things that are definitively in the background often appear in the literature is something I totally see as a big issue. And when you talk about the standardization, I presume you're not talking about any one particular piece of hardware. It's more about the standard practices and SOPs to ensure you're getting a genuine on-breath compound. So I was wondering if you could provide the audience, any kind of thoughts you have on what those types of reporting standards should look like.
Yep. So interestingly, we've been in talks with a number of groups about coming up with best practices types of things. And the idea there isn't to limit anybody's creativity for sampling or coming up with the next best way. But the way that I look at it is, I just got in the breath research in the last 8-9 years, and I didn't know a lot, and there wasn't, and paging through papers, right? And so you learn a lot, and so people starting need a place to start. And that's kind of where I'm getting with standardization. If you're going to do it this way, a lot of people do it this way. If you're going to do it that way, a lot of people do it this way. No way is necessarily the 100% way that you have to do it. But here's where I would start if I were you, kind of idea. Getting started in the breath research field.
Yeah, and I totally agree. There's no kind of magic bullet bit of hardware or anything that, there's always going to be pros and cons using different types of approaches. And I think we here at Alison, we kind of went through exactly the journey you're describing. You kind of try something, you learn, you're like, oh my God, there's tons of stuff in background. How do you actually deal with that? And some of the metrics that we've been thinking about, I don't know if you're on any of the talks yesterday, but we look at the blanks and then classify a VOC as on breath if it's 3 standard deviations above the mean of the background in at least 50% of subjects. And that's quite a harsh criteria, actually. So one of the things that we debate quite a lot is in biomarker discovery, early on in pilot, you can be a bit more permissive because stuff can fall by the wayside when you're trying to validate it. So should you have very harsh criteria to determine whether compounds are on breath at the outset, or relax that a little bit more and let validation deal with it down the line?
Yep. So we have generally went with the more relaxed approach. Just for a lot of the discovery aspects we do, I don't have a high N most frequently, and so I got to get the most options to go forward with validation, and then that justifies those additional studies to then be able to do more target analysis and eliminate things. Essentially what we do, we would rather have a wide breadth at the beginning and then narrow it down, as you said, when we do a validation type of analysis.
Yeah. No, I think it's interesting. That's one of those ones where there's no kind of right or wrong. It seems, I think actually that criteria of how frequently the VOC needs to be present within the population, I think that's quite tricky as well, because you can hypothesize that something may be present in a disease cohort that wasn't, if you're building up your VOC atlas and healthy controls. And actually, one of the interesting examples that was brought up to us was methane, which is definitively on breath. One of the very few breath tests that's actually available on the market, but actually not everyone is a methane producer. So if you were to class methane as being on breath and must be present in greater than 50% of the population, it wouldn't be classed as on breath a lot of the time. And from a hardware perspective, so I agree that you don't want to limit creativity, but do you think things such as using cleaned air for the subjects to breathe in, are there certain things that you can do at the front that will help from a data quality standpoint?
Yes, so there is, but it's all related to your ability to control those things, right? So using the receiver, obviously you can provide clean air to the participant. When I put somebody on a treadmill, I'm stuck with whatever's in the room. And so accounting for that room and truly understanding the background, not only from the TD tubes, but also the room, you have to take all into account for how you're analyzing your data.
Great. And it sounds then, so, with the TD tubes themselves, we have 4 tubes, and we've tried to pressure match tubes as well to try and deal with the issue of variability of flow. But it sounds like the overall conclusion is it doesn't have that much of an impact. Is that a kind of fair conclusion?
So based on the results I have, that is the conclusion. Now, I will say that it was a small sampling size, that I believe more work should be done there. We are actually moving towards something similar to what Christina Davis's group has done. So you would hook your pump to a real-time flow rate sensor, and it does a real-time calculation for your collected volume. And so it turns itself off. It's basically a 3-way valve and a real-time flow rate sensor. And so that gives you a more consistent result from your bag every time, independent of the TD tube, because you're measuring that flow rate real. And so we have gone this approach, and we've made something that we've started using that's very similar to what her group has published.
And in terms of the materials themselves, sorbent materials, have you got any? So there's the packing of the sorbent in the tubes. Is there work you think that needs to be done within the field around novel sorbents as well to give capture of VOCs that aren't, easily captured with 10X and carbograph and those types of things?
Yeah, so we have a researcher that's here that does mesopore silica, and that is better suited for aldehyde. So I think you can work with different groups, especially he, I believe, works with Sapelco, and to come up with novel combinations for what he does. So he picks a lot of targeted types of things. So he gets those media made specifically to target those analytes of interest. And so we are generally in a discovery scenario. So I try to grab as much as I can, rather than pick certain things. That may or may not be the best approach. It's just how we've gone about doing it.
Great. There's a question here that's relating back to environmental VOCs and VOCs of dietary origin, and I might try and elaborate a little bit further on it, because I suppose there's two sequential questions. So there's how do you make sure a VOC actually originates from breath and isn't just background, but when a VOC is on breath, it can still have an exogenous origin or an endogenous origin. So you can have a dietary compound which is on breath but is exogenous, and those were, it's endogenous and on breath. And so there's a question around distinguishing between something that's environmental and something that's dietary. I was wondering if you could try to tackle that a little bit.
Yeah, so I mean, the easiest example is isoprene. It's one of the main components in the environment, right? So, but it is also one of the most routinely found things in breath. So, I think you just have to be careful. So the hard part and we're trying to do it currently, but it's very difficult, is VOCs and animal work and cell culture to really get where the origination of these compounds is super hard. One, species differences in what's produced. Then you got all the background that's associated with bedding and the animals. So animal work is super hard. Cell culture is difficult due to plastics and sampling without contaminating everything. And I know there are people that do it, so don't get me wrong. But where volatiles originate is a super difficult topic to really get after. And so that's where we get hung up a lot, right? Like, you can't take somebody's lungs out. You can't, do all these things to really get at where volatiles are coming from. So I don't have a great answer for that. I think the best way to do it would be take appropriate amount of background and try to either subtract or either somehow account for what you're observing elsewhere.
Yeah, because it's I love the example of isoprene. I use the example of acetone as well. So you've obviously got acetone in the background. It's one of the most abundant compounds on breath. So if you, knowing that, if you saw a compound like that come out and you were to try and class it as endogenous or exogenous, you can make the argument both ways. Now, it's easy in acetone because there's an endogenous component, but it becomes a lot more difficult for novel compounds when you can't have a defined origin there. And I suppose one of the kind of interesting distinctions is a VOC would need to be on breath for it to be a potentially useful biomarker, but you can have exogenous compounds on breath that can be clinically useful and valuable. Even if you think about exposomics type research, that's the name of the game. You're trying to find those types of compounds. And a lot of what we've tried to do is deliberately put probes into target pathways that are non-exogenous compounds. So I was wondering, kind of, given what you said at the outset, there's a lot of just challenges of trying to figure out the signal from the background. And I think what we've tried to do here at Alstone is pursue two approaches of, broad-based discovery and a more targeted approach of putting probes in. I was just wondering if you were willing to try and make predictions about, if you looked at discovery and endogenous only, we know there's not many breath tests out there. Do you think that strategy will lead to more breath tests in the next 5 years if standardization is addressed, or at least we make steps in the right direction? And if so, what are the kind of disease areas where you think endogenous has the best role to play?
All right, so there's a number of questions there. So the first verse, on breath. So I feel that a lot could be done with isotopically labeled. Isotopically labeled glucose, where then you could look in your spectra to see where there are multiple peaks that correspond to shifts in mass, is a potential option to really get at endogenous versus exogenous, right? Your exogenous compounds would not be labeled. Now there are IRB and ethical issues that go into that, so we have not pursued that for those reasons at the moment, but that doesn't mean that it's not a viable option. So I mean, that is, the path forward that we are seeing for potentially truly determining what is endogenous and exogenous, right? You can see those shifts in mass and the endogenous shifts, or the endogenous peaks as well. So you would get same retention time, multiple peaks of mass in your spectra. That doesn't mean that that's the only option, but that is a viable option for truly honing in on what is endogenous versus an exogenous type of compound. I don't think that we're there yet.
Yeah, interesting. And what kind of disease areas do you think are likely to yield with breath analysis?
So there are many that options that you see the data come out, and it's pretty convincing, right? A wider range. So you have liver disease, you have lung cancer, all the asthma data is pretty convincing. So there are a lot of diseases. I don't know that one or two. I don't know that I would pick a one or two. I think they all have realistic options to work, and the more people investigate those disease states, and I think the better the data and the analysis will become, and more possibility for a clinical transition.
Great. And on the standardization side of things, so background and contamination has obviously been mentioned there. What are some of the other elements that you think need to be harmonized and standardized within the breath field?
Yeah, so we have done a lot of work on food. Not only we are working on developing a library of food-related volatiles, and so the idea there is we take the headspace above food. While I understand there's metabolism that's plausibly involved with all of that, oral microbiome, a lot that could go on there, and we're trying to make a database of food-related volatiles, so kind of headspace analysis. And then we also have very similar, where we will give individuals those foods and see what volatiles are present, and see how long they persist in their breath, right? And so the idea is we have historically used 1 hour of fasting as our rule of thumb. Obviously, more the better, but that is essentially what we've used as a rule of thumb. I'm not familiar with any distinct data that suggests maybe it's 15 minutes, maybe it's 4 hours. It probably depends on the food that was eaten. So the idea would be that we would have this library to say, oh, well, you told me you ate an apple half an hour ago. You should be good. I have data that suggests that all of the apple-related volatiles are below the background or something to that nature, is where we're trying to go with the contamination thing. Because we have people routinely where we keep them up for a very long time, and I can't limit them from what they can eat, but I can know what I could plausibly expect from what they ate, is the idea where we're trying to get. Because a lot of times, I'm very similar to the previous speaker. You don't have any control over eating and drinking. You can't stop the person.
Yeah.
And so knowing what to expect could help determine what you are actually looking at from their data.
Got it. There's one question here I thought was quite interesting as well. It's asking if you could blow directly into TD tube, would that help? No, we both know you can't effectively do that into TD tube, but you could plausibly have a different geometry of tube where you could blow directly into it. So if you had a sorbent material, obviously the sorbent tubes that we all use in breath research weren't really designed for breath research, but you could take a step back and say, okay, you could use a sorbent-based approach, change the geometry, and have a more direct sampling. I thought that was an interesting question. I was wondering if you had kind of any thoughts on whether or not that would be useful to the breath field.
I mean, it could be. It depends on. Again, it's similar to you do what you need to do to be able to sample properly within the constraints of your individual. And so I could envision a scenario where that might be useful. We've just never explored it, so I'm not, truly sure.
Yeah, we've done a little bit of work, but only in the context of very targeted analysis. So where you're looking for a single isotopically labeled compound. So yeah, you don't care about anything else necessarily in the background. Excellent. Well, I think we're coming up to time now. I just wanted to say a massive thanks again, Sean, for taking the time out to share the data and insights today.
I appreciate it.
And we'll share a link as well to all the participants for the JBR call as well. And so you said papers to the end of the year. What's the kind of expectation for when it would all come together then?
So they are rolling. As they come in, when they're accepted, they become posted and published. So it's a rolling. I believe you can submit until the end of the year, but then it's rolling until we have no more review.
Fantastic. I'm sure there will be many to review for many years ahead. And I think it'll be fantastic for the whole community to see those papers as they come. Thank you so much again, Sean, and have a great rest of your day.
Appreciate it. Talk to you later.